Yeah, well, actually, thank you for this talk, because actually it's a very good transition to the talk I will be giving about using siRNA as a therapeutic agent, potential therapeutic agent. And of course, as you know, there is a big question today, and this question relates to delivery of those double-stranded RNAs into human cells, into tissues, into organisms. And actually, I think we have to deepen our discussion afterwards as to the possibility to use them, to use your diabodies as peptides to deliver so the siRNA selectively and very specifically to living cells. Now, I would like to present some work that has been done at riboxx on the inhibition of rhinovirus, in this case in vitro and in vivo, using small interfering RNA. And so just a small agenda. Rhinovirus causes a common cold, Schnupfen in German. I would like to give some very brief overview on the epidemiology, the distribution, and the causative agents of common cold. Then, of course, directly go to the human rhinovirus. Here a small overview on molecular virology. I know that a lot of you, or many of you, don't like this, but I think it will be interesting to know how the virus replicates in order to understand and to predict how to target the viral genome and develop drugs to inhibit this virus and treat the common cold. And then we have two working hypotheses that we are now trying to verify. The first one, is it possible to inhibit in vitro the replication of the virus? And further on, based on those results, on this knowledge, is it possible to inhibit in vivo in animal models replication of the virus? So very briefly, the common cold is actually a neglected disease. Each one of us will have in the winter time an infection with a rhinovirus or a coronavirus, but more specifically a rhinovirus, and we'll have for let's say 7 to 10 days, maybe 2 days, maybe 3 days, but let's say 7. After 1 week, you are getting rid of the virus. This is actually a dogma for all RNA viruses like influenza or rhinovirus or P. coronavirus in general. Now in the US, there were about $4 billion that were spent per year, or that are spent per year, on medications for the common cold. So you have really a wide distribution of the disease with a huge impact also on the population, but also an industrial interest in developing drugs against this agent. But of course, although the infection is usually benign, you have also complications that occur, like asthma in children that will develop. I'll show you some slide. Later on, asthma, they will be exposed to the virus very early, develop a kind of hyperreactivity, and then further on a hyperactivity against other agents, sinusitis and otitis media. And of course, the major causative agent of the common cold is the human rhinovirus. So the infection, and this is important for the animal model that we'll be going through afterwards, occurs through a direct inoculation here in the rhinopharynx and goes then directly to the lung. So it's not only located there in the rhinopharynx, oropharynx, but it goes there downstream to the lung. And usually you have a viral clearance that occurs after a week, like influenza virus infection, 1 or 2 weeks. This is an RNA virus replication cycle. But you can also develop further on problems like or complication like asthma that, well, may lead to a kind of hyperactivity with this so-called airway remodeling. Now we know today that actually there is also a predisposition, an atopy predisposition to develop asthma in children that will experience this viral wheezing illness, really not really quite clear determined infections, which kind of agents do they to cause this viral wheezing illness. It's a virus, it's true, it's a human rhinovirus, it's true, but you have also to have this predisposition, and then in adulthood you will have this acute exacerbations with airway obstruction, wheeze and cough that will occur. So we have an infection that is really happening very early in infants and then leading in adulthood to complications that are really a burden also, a public health burden, but also a burden for each individual. Rhinovirus infection is also quite frequent. We just notice it. Each one of us will have maybe or probably an infection in winter. But if you compare, for example, this was done in France, in Lyon, 2008, 2009, in the autumn and winter time. So it's a seasonal infection. If you compare the number of infections with rhinoviruses in comparison, for example, to influenza. Here in blue, the flu, you have comparable, if not a little bit higher, number of infections with rhinoviruses in comparison to the common cold, to the common flu. And here, of course, RSV, respiratory syncytial virus. We have seen it and observed it each year as I was now working in the virology in the lab at the faculty. We have seen really each year those peaks on RSV infections were occurring in infants here in Dresden. So it's a quite frequent infection, actually. So now looking at the virus more precisely. So here's the virus. Actually, it's much, much smaller than this. It's a picornavirus. Pico from Greek means very, very small. Picomole, and so on and so forth. So it's about 28 to 30 nanometers in diameter. It's a virus without an envelope, so it's a naked virus. It's a single-strand RNA genome, and this makes it really interesting for a small interfering RNA targeting, because, as you know, you're targeting RNA, with a small interfering RNA. So you have now a perfect, let's say, at least from the chemistry, a perfect target, the single-strand RNA genome, but you have the problems that you have. So it's about 100 serotypes, a very broad family of viruses with a very high variability. At least the variability occurs here in the outer surface of the virus. This was also a problem, for example, by developing antiviral drugs against rhinovirus. There was this drug called pleconaril that is actually targeting the canyon of the virus. That is a depression that you have here inside or behind this protein here. And because of the variability, you couldn't really target all those 100 serotypes that were circulating, that are circulating. So there is a need for let's say, something going beyond the classical small drug development, something that will really target many serotypes in the genome, the mirror serotype of the virus, and small interfering RNA may be an answer by targeting the genome of the virus. So this is a genome. It's a single-strand RNA. It's like a messenger RNA. You have a cap-like structure at the beginning, a single strand, positive-oriented, and a poly A tail at the end. And this genome will be directly translated in the cytoplasm of the cell on the ribosomes to lead to giving a polyprotein. This polyprotein will be then autocatalytically cleaved by proteases of the virus and give then rise to the different viral enzymes. So basically, looking at the replication of the virus stuck here on a receptor. It's an ICAM1, for example, or an LDL receptor. Then you have this release of the genome in the cytoplasma, a naked genome, translation on the ribosomes, and then you have those proteins that are being then set up in the cytoplasma that will go then into vesicles. And you have cells infected with rhinoviruses, but also RNA viruses in general, a complete rearrangement of the Golgi. Everything in the cell in cytoplasma is going towards replicating the viral genome. There is a hijacking of the protein machinery of the cell by the virus, actually, by the viral proteins. And this, in those vesicles that are quite similar to lipid droplets, as a very high content in lipids, here and there you have replication of the genome, positive strand being transcribed in negative strand, and so on and so forth. And at one point in the replication cycle of the virus, you have this release of the virus that will infect the next cell and the next epithelium, the next tissue, and the next organism. So it's quite easy then to see that if you want to target the virus using small interfering RNA, you just have to look where you have the genome that is being naked, that is being then present to be targeted by RNA interference, and it's actually at the beginning and also in those vesicles. So our working hypothesis was, as we started working on this, is it possible at all to inhibit rhinovirus replication in vitro with small interfering RNA? We didn't know it at that time. First of all, when you want to do this, you have to think about a design. How will it look like, my small interfering RNA? Will it be 19, will it be 25 or 23? Which parts of the genome do I want to target? It's the beginning, it's the end, it's in the middle. And there are a lot of algorithms here, you can see them, that will allow you to give a kind of guess of a small interfering RNA of a given length with a given design. Now we have developed at riboxx our own prediction tool. So riboxx is RNA prediction tool that you can find at our homepage. And this prediction tool actually takes into account the following philosophy. You are using 4 different algorithms that are giving you 4 different information. The first information is which sequence is most suitable to be targeted. Does it have a very strong secondary structure, or is it quite a flat sequence? The second information is, how does it look like for the siRNA by itself? Do you have a very strong secondary structure in the small interfering RNA? You are dealing here with RNA, ribonucleotide, and they are very tightly packed. So if you have a very tightly packed small interfering RNA, you will have problems to release the strands that are of interest for you, and then in the next step to have them hybridizing on your target to initiate the RNA interference. And the last question, that's the algorithm we're also addressing, was the question of the free energy, of the folding energy of those small interfering RNA. So it was not only the primary sequence, it's also the 3D folding. And this algorithm that is on the web page, you can use it to for each messenger RNA you need. It's, for example, here we use it for musculus polymerase track. We use it for the siRNA that we designed in collaboration with Dr. MZU. We use it with other siRNAs for other scientists that are asking for it. And it just gives you then a result that is based on the congruence of those 4 or 5 algorithms. And the highest congruence will give you the highest score. This is the first siRNA, and so on and so forth. Now, this is the viral genome here depicted, and we have designed about 19 siRNA that are really were distributed on different parts of the genome and looked at their effect. And at riboxx, we developed an additional innovation, and it relates to the design of the siRNA by itself. So the classical, let's say, Tuschl-based small interfering RNA looks like this. You have 19 nucleotides, double-stranded 19 nucleotides, and at the end of the sense strand, or non-guide, but it's a 5' 3' sense strand, you have 2 overhangs, 2 nucleotides that are in overhang. It could be a uridine, it could be also a thymidine, so it could be DNA. On the opposite strand that is complementary to this one, you have also those 2 overhangs here that you can see it. Most importantly, the active strand in the small interfering RNA, as you know, is a guide strand or antisense. Here is the seed region. This is a region that will hybridize to your target messenger RNA. And here, cleavage will occur with an enzyme called argonaude-2, for example. There are 5 of them, but the most commonly used, known is argonaude-2. So you will have on the opposite strand, that is the messenger RNA, a cleavage at this position after the seed sequence has recognized the complementary RNA. Now, the reason why you have those 2 overhangs is quite easy. The reason is that, according to the rules of El-Basher and Tuschl, you need a turbodynamic instability in the small interfering RNA. You need here to have a more loose binding of the double strand in comparison to this part of the small interfering RNA in order to allow opening of this double-stranded complex. Now, we then had the idea to say, okay, let's just get rid of this kind of voodoo, how to design it with overhang, without overhang, and so on. Let's try to get some standardization. And the standardization that we brought was to say, we're just having blunt small interfering RNA without overhangs, and at the end, we induce this thermodynamic instability in adding a GC clamp, 5 Gs, 5 Cs. And we tested this. And also the results that were shown in the first talk by Dr. MZU were using this design of riboxx.